Good afternoon. Can you hear? Thank you. Okay, so we will start with last time we had seen nucleic acid databases. Now we'll turn to protein sequence databases. So this protein sequence databases is the collection of data, and this data is collected after the study of physical, chemical, biological characteristics of the protein, its structure, its function, its three-dimensional structure, its interaction with other proteins. So all these characters are taken into consideration during the collection of data. So when this data is collected, it is introduced or it is added or updated in the protein database. So this collection of data involving physical, chemical, biological characteristics, structure of protein, function of protein, three-dimensional structure of protein and its interaction with other proteins all will be included in a protein database. So it may form a protein sequence database. Okay. The biological information of these proteins is available in the form of structures and sequences. These sequences, they are represented uh, in the form of three-dimensional structure or a three-dimensional data of sequences. This protein database has one or more data sets about the proteins and which include the amino acid sequence of the protein, the conformation of protein, the structure of the protein, and the features of the active site of the protein. हे सगळे त्या डेटा सेटमध्ये अवेलेबल असतात अँड असे अनेक डेटा सेट्स एकत्र येऊन तो प्रोटीन डेटाबेस तयार होतो. Several protein sequence databases are available and they include primary databases like Protein Information Resource PIR, Protein Sequence Database PSD, SwissProt, Protein Data Bank, etc. So these are the primary databases. Now what is SwissProt? This is a well-known and most extensively used database so far as proteins are concerned. Curated protein sequence database. It is nothing but a curated protein sequence database and it provides high level of annotation. Means lot of information is stored in this type of database and high level of information is stored in this database. Just like protein information resource, that is PIR, and protein sequence database, that is PSD. Uh, this data is updated or added in SwissProt as core data and annotated data. The core data, it consists of sequences entered in a common single letter amino acid code. Okay, कोडिकल फॉर्म मध्ये अमायनो ॲसिड्सची सिक्वेन्स टाकली जाते and related references and bibliography is also added. So the taxonomy of the organism from which the sequence was obtained was also a part of this core information. Core data मध्ये काय काय येईल? Amino acid sequence, ती अमायनो ॲसिड्स सिंगल लेटर कोडच्या फॉर्ममध्ये टाकलेले असतात. त्याच्याशी कनेक्टेड रेफरन्सेस आणि बिब्लिओग्राफी किंवा जोही डेटा कुठून कलेक्ट केलेला आहे त्याचे रेफरन्सेस हे सगळे त्या कोर डेटामध्ये इन्क्लुडेड असतात. तो प्रोटीन कुठल्या ऑर्गॅनिझममधून आयसोलेट केलेला आहे, तो ऑर्गॅनिझम कुठल्या जीनस स्पेसिसचा आहे, हे सगळे टॅक्सोनॉमिक कॅरेक्टर सुद्धा या कोर डेटामध्ये इन्वॉल्ड असतात. Then annotation consists of information on function or functions of protein. So annotation मध्ये काय include होईल? Core data includes sequence of amino acids, or it includes the taxonomical features of the organism from which the protein has been isolated. What does the annotate data include? It includes the applied aspect, that is a function of that protein, the post-translational modification in that protein like phosphorylation, acetylation, glycosylation. आपण हे सगळे post-translational modifications, we have seen them. Then functional and structural domains in this protein such as calcium binding regions, ATP binding regions, zinc fingers, etc. Secondary features such as alpha helical, beta sheet, quaternary structure, means alpha helical structure of protein, beta sheet structure of protein, quaternary structure of proteins, similarities between proteins of various organisms, diseases that may arise due to the protein deficiency of that protein or excess synthesis of that protein, uh, mutations in the genes involved in the synthesis of those proteins. So, resulting mutation may cause which type of genetic problem, which type of mutation, and that mutation may lead to which type of disease. All this will be included in the annotation. So he protein sequence database, it has been grouped into two. It contains two types of data, the core data and the annotate data. The core data will include the sequence only and from which organism it has been isolated, or where is the taxonomic position of this organism with respect to other organisms at genetic level, at species level, etc. To which order they belong, whether they belong to yeast, whether they belong to bacteria, eubacteria, archaebacteria, etc., etc. So that will be included in the core data. In the annotate data, you will have lot of information. So that information will be the function of the protein, the post-translational modifications that are involved in the protein, or whether there is acetylation after protein synthesis or glycosylation. हे सगळे त्याच्यात include होईल. Then which type of binding sites are available in that protein for interactions, protein-protein interactions. Then what is the secondary structure of these proteins, or like alpha helical structure or beta sheet structure or quaternary structure. How this protein is similar to other proteins or dissimilar with respect to other proteins. The diseases associated with the uh, extra synthesis or less synthesis of that protein, the gene involved in the synthesis of that protein, mutations in the gene may lead to what type of problems, that each and every data will be included under the annotated data. Now, translated EMBL. What is this? Trimble is a computer annotated protein sequence database that is released as a supplement to SwissProt. SwissProt मध्ये कोर डेटा आहे, अनोटेड डेटा आहे, त्याच्यासोबत ट्रान्सलेटेड EMBL पण आहे. So what is that? It is nothing but a computer annotated protein sequence database, and it is along with the other data like core data, annotated data, etc. In addition to that, translated EMBL is also a supplementary part of SwissProt. What does it contain? It contains translation of all coding sequences present in EMBL nucleotide database. So where in protein nucleic acid databases, we have seen EMBL nucleotide database. Okay? म्हणजे what is the sequence of nucleotides? on the DNA associated with the synthesis of a particular protein. So this has been supplemented with Swiss prot, which is involved in the storage of protein sequences, or it is nothing but a protein sequence database. So that database is complemented or supplemented with nucleotide sequence database. Okay, and in nucleotide database, this information is not there, but translated EMBL has this information that this sequence of nucleotides is involved in synthesis of this protein. So it may contain sequence of proteins that have never been expressed and identified in organisms. So Tremble will include everything, including sequence of nucleotides involved in the sequence of proteins. In addition to that, sequence of nucleotides which is never expressed, or we do not know what type of protein it has been involved in, but it has been identified in organisms. Then protein data bank. This protein data bank is a primary protein structure database. It is a crystallographic database for three-dimensional structure of large biological molecules such as proteins. This PDB is a primary structure database, and crystals of these proteins, crystals एक्सरे क्रिस्टलोग्राफीने एखाद्या प्रोटीनचं क्रिस्टल स्ट्रक्चर स्टडी केलेलं असेल किंवा लार्ज बायोमॉलिक्युल्सचे प्रोटीन क्रिस्टलोग्राफिक स्ट्रक्चर स्टडी केले असेल तर त्याची इन्फॉर्मेशन ह्या पीडीबी मध्ये स्टोर्ड असते. PDB archives the three-dimensional structure of not only proteins but all biologically important molecules. It doesn't include only proteins. It includes other biological molecules also, such as fragments of nucleic acids, RNA, peptides, etc. Then there are certain complexes of DNA with proteins. We know that nucleic acids may be associated with proteins. So this complex of protein and nucleic acid, the sequence of such structures or this interaction between the proteins and nucleic acids, किंवा त्यांच्यामध्ये काय कसा स्ट्रक्चर निर्माण होऊ शकतो प्रोटीन आणि न्यूक्लिक ॲसिड्स हे कंबाईन होऊन किंवा प्रोटीन DNA इंटरॅक्शन्स तयार होऊन कोणते स्ट्रक्चर्स तयार होऊ शकतात त्याची इन्फॉर्मेशन सुद्धा या PDB मध्ये 3-dimensional फॉर्म मध्ये अवेलेबल असते. The protein data bank is a database for 3-dimensional molecule structure of the large biological molecules. So 3D structure असेल large biological molecules तर ते PDB मध्ये तुम्ही स्टोर्ड असं फक्त एवढंच तुम्ही बेसिकली with respect to your studies you have to remember what is PDB or what is Swiss prot. So Swiss prot you remember core data and annotate data. What is the supplement of Swiss prot? Tremble is the supplement of Swiss prot. What is it associated with? Okay, Tremble is associated with those sequence of proteins which have not been expressed or not identified in microorganisms. What is protein database? Protein data bank, protein data bank involves, it is nothing but a crystallographic database. So it involves 3D structures of various type of macromolecules or biological molecules. These structures, they can be obtained by different techniques which you have studied in MSc Part 1, X-ray crystallography, nuclear magnetic resonance spectroscopy, cryo-electron microscopy, and they have been submitted by various type of researchers, biologists, physicists, biochemists. They have been dealing with such type of molecules and they have studied the structure of these molecules by any of these techniques, and whatever data they obtained they have submitted to the protein data bank, and that has been made available to people, interested people all over the world through this protein data bank. It is freely accessible by internet and its member organizations are PDBE, PDBJ, RSCB and BMRB. PDB is overseen by an organization known as World Protein Data Bank. So if you go to www.pdb.org, you'll be able to see what is this protein data bank. And World Protein Data Bank is the organization which controls this protein data bank. PDB is key, or it is involving the structural biology part as well as structural genomics. फक्त structural biologist नाही, but genomics पण त्याच्यात involved आहे. Most major scientific journals and funding agencies require scientists to submit their structure to the PDB. We should have that knowledge. Even if you have the structure of protein, you need to have the knowledge to submit that data to PDB. Many other databases use protein structures deposited in PDB. For example, SCOP, CATH classify protein structures, while PDB-Sum provides a graphic view of PDB entries. So काही ठिकाणी स्ट्रक्चर फॉर्म मध्ये असतं, काही ठिकाणी PDB-Sum आहे. So that involves the graphic information. And जीन ऑन्टॉलॉजी मधून ती इन्फॉर्मेशन कलेक्ट करून तिथे सबमिट केली जाते. द डेटाबेस होल्ड्स डेटा डिराइव्ड फ्रॉम मेनली 3 sources: structure determined by X-ray crystallography, NMR experiments, and molecular modeling. So whatever data is collected, it is collected by passing the protein through these techniques, or by analyzing the structure of these proteins by these techniques, and then that data is submitted. The PD database is updated weekly. आठवड्यातनं म्हणजे बुधवारच्या दिवशी त्याला अपडेट केलं जातं. And according to 1st April 2020, I have that only information. X-ray diffraction किंवा X-ray crystallography किती प्रोटीन्स त्यांनी आयडेंटिफाय केलेले आहे किंवा डेटा बँकमध्ये अवेलेबल आहेत. 1,35,170 proteins, 2,097 protein nucleic acids. Protein nucleic acid complexes are 6,945, 6,945. There are some other type of data of various macromolecules, that is 4 unidentified part. So totally, how many proteins have been reported by X-ray diffraction in PDB, or they are available in PDB? That is 1,44,260. By NMR, 11,337 proteins structures have been studied, and that data is available in PDB. Nucleic acids 1,325, complexes 264, and other 8. So total 12,934 proteins or their complexes or nucleic acids, their data has been submitted to PDB, identified by nuclear magnetic resonance spectroscopy. Then electron microscopy ने काही आहेत, काही hybrid methods आहेत. So 2 or 3 methods have been used together. So those hybrid methods ने सुद्धा काही प्रोटीन्स आयडेंटिफाय केलेले आहेत, आणि ते आहे 155 proteins, nucleic acids 5, protein nucleic acid complexes 3, other 1, and total 164. So if you see a total, it's 1,50,423 proteins are available, and their information is available in the PDB database. 3,466 nucleic acids have been reported in protein database, and totally 1,62,269 proteins or nucleic acids or protein nucleic acid complex database information or data is available in PDB. So this PDB, it includes UniProt. It's freely accessible database of proteins. That is a website. If you go to this website, you can access the UniProt protein database, and it includes the functional information of this protein. The information in this database is available through genome sequencing projects. It contains large information about the biological function of proteins, which has been reported in various types of writings or journals or research papers. It is maintained by UniProt consortium, which consists of European bioinformatics organizations and a foundation from Washington DC, United States. हे दोघं मिळून त्याला सांभाळतात. UniProt is useful in finding protein sequence and functional information. It is useful for inferring evolutionary information. So if you refer to the UniProt, you'll come to know how this microorganisms have evolved, or the proteins of that microorganism, those microorganisms have evolved. UniProt provides 4 core databases: UniProt KB with subparts SwissProt, Trimble, UniPark, and UniRef. Then next one is UniProt KB. So it is UniProt knowledge base. It's a protein sequence database partially curated by experts consisting of 2 sections, UniProt KB or SwissProt. All manually annotated entries are there, म्हणजे कुणीतरी ते एंटर केलेले आहेत. UniProt KB and Tremble contain unreviewed automatically annotated entries. So in UniProt KB or SwissProt, the data has been entered by some experts who are handling this UniProt KB, or some of the data has been automatically added through computers or through internet. UniProt KB or SwissProt is manually annotated non-redundant protein sequence database. याच्यामध्ये रिपिटेशन्स नसणारे म्हणून त्याला नॉन redundant म्हटलेलं आहे. And it is manually annotated. So information is added manually. It combines information extracted from scientific literature. So somebody must be referring that scientific literature आणि तिथून ती information collect करून ती manually update केलेली आहे. Computational analysis is also used for that purpose. The aim is to provide all known relevant information about a particular protein. So those who want to study these proteins, they can refer UniProt KB and get that information. Sequences from the same gene and same species are merged into the same database entry. Differences between sequences are identified and their cause documented. What is the difference? का difference आहे, काय difference आहे आणि काय आहे. So that is also noted there. For example, alternative splicing मुळे ते डिफरन्स असू शकतं. नॅचरल व्हेरिएशन मध्ये म्हणजे एका ऑर्गॅनिझम मध्ये हा प्रोटीन आहे आणि त्याच्यापेक्षा नंतर डिस्कवर झालेल्या ऑर्गॅनिझम मध्ये तोच प्रोटीन्स काहीतरी व्हेरिएशन दाखवते. So that is because of alternative splicing method, you know, we have studied it, or because of natural variation or incorrect initiation sites or incorrect exon boundaries or frame shift type of mutations or unidentified conflicts. So there may be many reasons why there is difference between those two proteins of the similar type or involved in a similar type of role or function. So even if they are same proteins, why there is a difference? What is the gene is same, still the protein synthesis are slightly different. What is the reason behind that? That has also been incorporated and documented in UniProt KB or SwissProt. Then UniProt KB Tremble. It contains high quality computationally analyzed records. So this record has been added through computer management or computer analysis, and it is automatic type of annotation. So nothing is done manually. Automatically things are collected from various sources and incorporated in this type of database. It was introduced in response to increased data flow resulting from genome projects. Several people are carrying out genome projects and lot of data is generated, and it becomes very tedious or time consuming for the manual annotation. So if anybody is going to add it manually, lot of time will be required for that process. So better it is done automatically. So automated things will not require any type of manual work. ऑटोमॅटिकली त्याच्यात ॲडअप होत जातील आणि म्हणून हिची निर्मिती झालेली आहे. इन SwissProt it had to be done manually, but in Tremble it is done automatically. The translations of annotated coding sequences in EMBL Bank, GeneBank, DDBJ, न्यूक्लियोटाइड सिक्वेन्स डेटाबेसेस आर ऑटोमॅटिकली प्रोसेस. बघा आता हे कनेक्टेड जरी प्रोटीन डेटाबेस असेल तरी जीन बँक is connected to it, DDBJ is connected to it, EMBL is connected to it. So some data from there correlating with the proteins will be automatically updated. Like this is the nucleic acid sequence, this is associated with this type of proteins. All this will take place automatically in Tramble. UniPark, it's UniProt archive. Short form is UniPark, is a comprehensive and non-redundant database. So all is compact in this type of database. It contains all protein sequences from publicly available protein sequence databases. So UniPark is also a very good means for getting information if any researcher, a new beginner is involved. Proteins may exist in different source databases in multiple copies in the same database. In order to avoid redundancy, UniPark stores each unique sequence only once. So it may be possible that a researcher from India is doing the same sort of research and submitting the protein sequence or its role or a function or some type of characteristic feature to the PDB, protein database. Some person from France is doing the same type of work, so there will be repetition. That is known as redundancy. To avoid that redundancy, UniPark has been generated. So it has if there are two data available of the same type, that will be introduced only once. Identical sequences are merged regardless of whether they are from the same or different species. So if the sequence is identical, it will be noted that these two sequences from different species are identical. Whether it is a microbial species or a bacterial species or yeast, this sequence is similar in both the cases. So it will be introduced only once in UniPark. Each sequence is given stable and unique identifier. So UPI code will be given, and that code will help you to tell whether it is from bacterial source, viral source, yeast source, but the sequence is similar, making it possible to identify the same protein from different source databases. It contains only protein sequences with no annotation. So only त्याच्यामध्ये sequences आहेत. Is that clear? Next, scoring matrices. So sequence alignment and database programs compare sequences as series of characters. Okay, different types of sequences आहेत. Protein sequences, nucleic acid sequences, and various types of database programs. They compare these sequences as series of characters. ह्यांच्यामधलं comparison, sequence कशी aligned आहे, in which direction it is, in which polarity it is, ह्या सगळ्या जी इन्फॉर्मेशन आहे वेगळ्या वेगळ्या सिक्वेन्सेसची, न्यूक्लिक ऍसिड असो की प्रोटीन सिक्वेन्सेस असो, सो दे त्यांच्यामध्ये कंपॅरिझन कसं केलं जातं ॲज सिरीज ऑफ कॅरेक्टर्स. All programs for comparison are based on some scheme, and that scheme is known as scoring scheme. Scoring matrices are used to assign a scores to each comparison. If you have compared two proteins or protein and nucleic acid or two nucleic acids, then some score is given to it, to that comparison, and that is calculated by scoring matrices. These scores are nothing but integer values. A positive integer means similar characters and negative integer means dissimilar characters. So if you get a positive integer, it means that the characters are similar. If there is a negative integer or if the characters are different between the two proteins, you give it a negative integer. Identity scoring. So there are different means of scoring. It's a simple scoring system. The identical characters are given score 1 and non-identical characters are given score 0. वरती काय होतं? स्कोरिंग सिस्टीम मध्ये पॉझिटिव्ह इंटीजर सिमिलर कॅरेक्टर्स, नेगेटिव्ह इंटीजर डिसिमिलर कॅरेक्टर्स. आयडेंटिटी स्कोरिंग मध्ये इट्स स्लाइटली डिफरेंट स्कोरिंग सिस्टम. आयडेंटिकल कॅरेक्टर्स स्कोर 1, dissimilar characters score 0. DNA scoring involves variations as transitions and transversions. DNA scoring मध्ये काय दिलं जातं? Transitions and transversions. So identical characters are given score 3, transitions are given score 2, and transversions are given score 0. Okay, transversion, yes, mutation study केलेले आहे आपण. Transition, purine replaced by purine. Transversion, purine replaced by pyrimidine. So if there is transversion observed in a particular sequence, म्हणजे A च्या जागेवरती तिथे C असेल तर it is transversion. It is a dissimilar character. किंवा आयडेंटिकल कॅरेक्टर्स असतील, एक सिक्वेन्समध्ये न्यूक्लिक ऍसिडच्या आणि दुसऱ्या सिक्वेन्समध्ये आयडेंटिकल बेसिस दिसून येत असतील तर द स्कोर इज गिवन ॲज 3. If there is transition, means if a purine is replaced by another purine, then the score is given by, like A is replaced by G, score is given as 2. If there is transversion, like A is replaced by C, purine is replaced by pyrimidine, it's a transversion, so score is given as 0. Now इथल्या इथे तुम्हाला 3 systems दिसून गेल्या. Identical characters, positive integer, dissimilar characters, negative integer. This is one system. Another identity scoring system is identical characters score 1, non-identical characters score 0. तिसरी system आहे identical characters score 3, transition score 2, transversion 0 score. Means it is totally dissimilar. Then chemical similarity scoring is also given. This scoring matrix consists amino acids with different group shapes and charge. त्याच्यामध्ये मग chemical similarity scoring involved असेल. Observed matrices, it is the most commonly used matrix for proteins. Substitution frequencies are taken into consideration in this matrix. So here, which type of amino acid is substituted, how many times? Like if there is histidine amino acid, आता हिस्टिडीन किती वेळा रिप्लेस झालेला आहे, किंवा ॲलनिन आहे तर ॲलनिन त्या जागेवरती किती वेळा रिप्लेस झालेलं आहे, तर दॅट सबस्टिट्यूशन फ्रिक्वेन्सी इज टेकन इन टू कन्सिडरेशन इन दिस टाइप ऑफ matrix, and it is known as observed matrices. The score in observed matrix is based on evolutionary occurrence of proteins. So evolutionary occurrence मध्ये डेव्हलपिंग ऑर्गॅनिझम्स मध्ये हे प्रोटीन्स कसे अकर होत आहेत, त्यांची सबस्टिट्यूशन फ्रिक्वेन्सी काय आहे, दॅट इज टेकन इन टू कन्सिडरेशन. It includes the frequency of occurrence of a particular amino acid in nature. It will give idea about abundancy of a particular amino acid in nature. Like अगदी प्रिमिटिव्ह ऑर्गॅनिझम्स मध्ये हे अमायनो ऍसिड्स खूप कॉमन आहे. त्याच्यापेक्षा हायली डेव्हलप्ड ऑर्गॅनिझम, बेटर डेव्हलप्ड ऑर्गॅनिझम मध्ये हे अमायनो ऍसिड्स कॉमन आहेत. त्याच्यापेक्षाही मोर डेव्हलप्ड ऑर्गॅनिझम मध्ये different amino acid is common. So that will give you a evolutionary occurrence of these amino acids and how frequently they are present. That idea will also be given by the observed matrices. Identical cases are given positive scores, dissimilar cases are given, or mismatch cases are given negative scores. The matrix gives the ratio observed frequencies of amino acid as to frequencies that occur by chance. So what is the observation? How many amino acids are occurring in a particular group of organisms or with respect to evolutionary development of organisms, and चुकून एखादा कस काय ते फ्रिक्वेन्सी तुम्हाला दिसून आली, बाय चान्स कुठला अमायनो ऍसिड अचानकपणे तुम्हाला दिसला, that ratio is given, and this ratio is known as odds score. The logarithm of this odds score, म्हणजे तुम्ही काय करायचे, ऑब्झर्व फ्रिक्वेन्सी ऑफ अमायनो ऍसिड काय दिसलीये तुम्हाला इव्होल्युशनच्या ट्री मध्ये, किंवा as organisms have evolved, which type of amino acids are frequently observed and which are occurring by chance. त्याचा रेशो काढायचा, त्या रेशोला आपण ऑड स्कोर म्हणतो. त्याचा लॉग काढायचा, तो लॉग असेल लॉग ऑड स्कोर्स. बोथ आर यूज टू स्कोर प्रोटीन अलाइनमेंट. याच्यावरून मग प्रोटीन अलाइनमेंट कॅल्क्युलेट केले जातात. सो देर आर टू matrices, PAM and BLOSUM matrices, which are commonly used, observe logs odd score matrices. यांना आपण ऑड्स स्कोर मॅट्रायसिस असं म्हणतो. आणि पॅम आणि ब्लॉझम हे दोन टाईप ऑफ मॅट्रायसिस आपण आता नेक्स्ट लेक्चरमध्ये बघणार आहोत. सो keep on reading all these things. Reading will help you to understand. We'll see it in the next lecture. This week we'll be finishing with everything, and I'll give you the notes simultaneously on Saturday. Okay, so let us stop here. If you don't understand anything, you can call me or ask me on chat. So you can also even send your personal chat to me if you have any difficulty. Let us stop here. Thanks.